we'll move on to the roundtable session with Michelle Jeanette. Once again, Michelle is the founder of Cure KCNH1 Foundation. Michelle, you with us?
Hi, everyone. Thank you again so much. These afternoon presentations were so exciting and absolutely fascinating, and we all love to hear about the amazing work you are doing in the space. So I'd like to start out with kind of a broad question. I know we have a lot of ASO people here today, but I'm curious about where you all see things in, all of us parents want it to be yesterday or 3 years ago or whatever, but where do you see the space in 5 years, specifically in terms of the roles of different modalities? All right.
I can start.
Jeff, I see you. There's Winston. Hi. Winston,
Yeah, so,
you kind of have an overview with the N of One Collaborative of some of the different approaches people are taking. Can you give a stab at that?
Sure, I'm happy to. And I think there are lots of things about drug development and its complexities that I will have to defer to Jeff and Irina about. But from my perspective, I think one of the things that is, I don't think there's going to be a one-size-fits-all solution, right? And I think that's because of the complexity of genetic disease, but also because of realistically the limitations to how we can get to some of the target tissues of interest. So what that means is if you take an ASO, right, a lot of the ASOs these days, we're thinking about the intrathecal delivery because we're building upon the foundations of Spinraza and all the great work done at Ionis. There's other ASOs that work, they're delivered systemically. Those can, Jeff is talking about the designs for them. So I think where you're thinking about different modalities, it starts off as trying to work backwards from what is the actual need of a given disease. If you correct a gene in the liver, will that have an effect on, systemic metabolic disease? Some diseases, yes, it actually really is impactful just to do that liver thing. Or even if your liver symptoms are treated, your brain ones can be alleviated sufficiently to give you quality of life, right? Other ones, the primary factor may be the neurodegeneration, and getting CRISPR into the brain is still an incredibly difficult challenge. So I'm not doing a great job of kind of making it simple for rare disease families to say, how do we navigate the space, right? I think I wish there were a more like, here, just try this. But I think that's where that partnership is really valuable. My last point is that I do think there's a, there will be these event horizons of having new modalities approved that will enable, just like, Spinraza enabled the imagination of an entire group of investigators, or rather Spinraza enabled Mealison, which then enabled more people. I think there's going to be that case with CRISPR and some of these N of 1 CRISPRs. And my prediction is that's probably going to happen in the blood space, in the hematopoietic stem cell or T cell editing space with CRISPR, just because it's easier to get and access those cells, check it for off targets, and then do the editing and reintroduce it. So you may see immunodeficiencies like inborn errors of immunity that are the first on that N of 1 as opened up to broader gene editing modalities. The last thing I know I just said one last thing, but the last thing is I think there's also the inherent long-term effects of gene editing, which is more permanent than RNA editing or RNA modulation with modalities such as antisense oligos that one has to consider. So it's complex, and I'll certainly open it up to anyone else, especially seasoned drug developers, for their thoughts on this.
Yeah, and I was just to follow up on that, Winston, the delivery part is a huge part of RNA targeting and RNA therapeutics in general. It's just it's almost like they have to go hand in hand. Every time you think about the disease you're going after, you have to think about the delivery component of that and making sure that you have a potent enough molecule to get to the tissue you want to work within a therapeutic index. But Spinraza did pave the way for us, our imagination, exactly what you said, Winston, and I feel like I'm following off what you say every time, just like it's validating. But in any case, the splicing landscape, I think, understanding splicing, understanding and getting enough data to understand that in a systematic way is going to open a lot of doors for a lot of diseases that we didn't know we could do. And so keeping an eye on, that's what we're doing at Loyal Labs. We're keeping an eye on that space and trying to understand it as well as we can so that we can design strategies where they weren't, because we didn't have access to that data before. So I think that's one area that's really exciting. And the fact that it does work in neuro tissue as far as delivery, that's a very compelling thing. But also to keep in mind that, when you look at splicing oligos, Ionis was not a splicing oligo company. So everybody should keep that in mind, because the modality that was used at Ionis had a shorter half-life. So the systemic studies were never really done. So there could be a lot of room, that still needs to be sort of explored a little bit as far as being able to do splicing in other tissues. It was never really studied, even though you think about Ionis as the one who were the experts there, they never did it. So there's still there's a lot of great potential with that modality that hasn't been explored yet.
And what about the other main ASO technology, the knockdown approach, whether that's allele specific or allele agnostic? Do we have any success stories like, obviously, I don't think there's any other success story like Spinraza, but good examples of great therapeutic benefit with a knockdown approach?
I don't know of any therapy that's out there. Oh, sorry, go ahead.
Go ahead, Jeff. Go ahead.
I was just going to say that I know that there's proof of concept data with siRNA from Exo Labs. And we're doing it here with RNSH mediated knockdown, as La Jolla Labs is partnered with and Lorem. So as a result, we're using gapmer ASOs for allele selective knockdowns. But as far as therapy, I don't think there's anything that's actually gone.
Hopefully working
I know
some
there
of our
isn't.
kids as we speak.
Yeah, awesome. Yeah.
Yeah, I just wanted to say that I agree that there's nothing like approved, and Spinraza really is, I think, it's just a wonderful story. But I think also there was luck involved. I mean, I think, at the time, this just really worked out well, which is great. It's great for everyone. But I think we cannot assume. I think we shouldn't assume that just the mechanism itself is kind of like a key driver of the success. I mean, I think for some of the clinical trials with knockdown, there are lots of factors that went into that. And I mean, I just personally, I just want to add, delivery is so important, delivering the drug to the target tissue. And so my personal bias, and I used to work on Huntington's disease for actually many years. I just think that if we don't get the drug to where it really needs to go, it's really hard to really conclude, other than that there was I think a safety issue in the Roche and Ionis trial. But I think it's not necessarily the mechanism. I just think that we need to get the drug to where it needs to go, and I personally do hope that over the next 10 years, there will be easier ways to administer even a naked ASO for a brain disease than intrathecally, because I think intrathecally we know that the distribution, particularly in the deeper parts of the brain, where I think for Huntington's disease it's important to get to, is just not very high, particularly with the, let's say, first generation ASO. So I think between improving maybe ASO design and then maybe finding a way to deliver, I think we will see some progress, and I'm excited to watch the progress. At Everyone Medicine, we are not investing in new delivery modalities, but we will be watching the field. And then the same with other modalities. I mean, I think in a first case where you don't know what happens when you sort of change splicing or knockdown, I think an ASO has the advantage that you can discontinue treatment. And if we have very, very few patients, so we don't have a large safety database, I do think this is a consideration that is in favor of ASOs rather than permanent modification, even if I understand that this might be desirable to have a one-time treatment. But I think ASOs have the advantage that we can discontinue if we see something. And I mean, Tim himself presented a case where, maybe this was good that we had this opportunity.
100%. That's a huge, huge advantage.
If I can add on 2 comments. One is, I think, again, thinking back to the modality, the genetic modality of editing, right? There are certain things that with ASOs right now you can't do in terms of correction back to the wild type, right? And this is where some of the gene editing technologies like base and prime editing could be advantageous, because one of the things that you always ask yourself with doing any sort of knockdown target or splice modulating target is like, well, this doesn't really exist in nature. Like, we don't have, human examples of someone who lives with a splice modulating oligo, but we do have the wild type of, billions of other people who, if you correct it back to the wild type, there's a therapeutic hypothesis there. So that is, in some ways, it can be a little bit better from the target safety perspective, but I completely agree with Irina that there's unknowns about the long-lasting duration of any off targets. On the other
Yes,
hand,
that's certainly what is so appealing about base editing, is just, correct to that healthy state versus knocking down either a full or 50% of a highly conserved gene.
Yeah, and I will say there are some RNA editing technologies coming online that might, enable that thing at the exon or mRNA level. My last comment is I think I flip it back to you, Michelle, of saying there's actually, in some instances, in ultra rare cases, there's so much that drug companies can also learn from N of 1 trials that are happening in actual people. And I really say this with all the humility and like sensitivity in the world, that we're not experimenting on patients, but when there is a risk benefit and you're like, we have to do this, and you have an investigator initiated trial, and they might be trying a new technology that's gone through all of the, the FDA, IND approval processes. There's a lot that can be learned when it's spearheaded by, individual foundations or families, that the risk of not treating is worse, right? And I think in those cases, it's important to know that the value that's from one in-human trial can be like more than hundreds of, monkeys or rats or whatever. And there's a place where I think sharing that information and being the intrepid explorers there is so valuable to the entire community.
Yeah, absolutely. And to those of us who are in the space doing this, well, that kind of leads right into my next question about the risk benefit analysis. I think people out not living our lives look at it and say, oh, your kid's pretty stable. Why would you, XYZ? But for those of us living with these rare conditions, we're used to living with a lot more risk than most other people experience with their kids. Every little minor cold could be a big risk to any of our kids. Every day there could be a seizure. We're used to a lot more risk in our lives. And so I think we're eager to take prudent risks to advance the science, and absolutely sharing those learnings are crucial.
I think one thing we think about a lot is partnering with patients and the families to make sure we're capturing the benefit in the most meaningful ways as well. Because
Yes.
I think there are, clinical outcome measures, and there's like disease
Worthless.
constant models. A lot of these things, I think, they lack the nuance when it comes to how it's impacting your life. And so
100%. Tristan, for example, it has been rated in the less than 0.1 percentile cognitively and adaptively. So I can, pump him full of moderately effective and even potentially life-changing drugs all day, and it might still not move the needle on the Vineland, for example. Like there's a major floor effect with our profoundly impaired kids.
And Julia mentioned things like, when Millicent was administered and, coming down on some of the anti-seizure medication, Mila was able to sit more upright. And that tiny little thing that wouldn't be picked up on any outcome measure that a doctor sees means that, she wasn't always slumping over, right? And then potentially choking on things, or, cleanup and all of that. The little things that affect one's life just are so important to capture for that benefit. And it's not always just about the risk, right?
Yep, absolutely. I think, you guys saw a bunch of little clips, vignettes in the KCNH1 community video about the little ways that me out before, never would have thought of rare disease affecting your day-to-day life. And most of those measures don't even ask about something as huge to us as like potty training, for example, just some seemingly little and may not be measured on outcome measures could just be hugely impactful in terms of quality of life.
And maybe I just say something for whatever it's worth. Sorry, it's so in my past job, I mean, where I said like I was working on Huntington's disease, some of the regulatory agencies were absolutely adamant that we have actually discussed outcomes with affected families and patients. Because even though maybe you have your standard scale that might not move much, but that you have something else that is really relevant. And some regulatory agencies are more open to that than others. But I think making sure that those who are not that open, that when we go to them, that we have, somebody representing, particularly for NO1, maybe, that particular patient or that family, or, I think it's very important so that they understand why this is so important when it maybe looks like not so important to a regulator and maybe even to a clinician, So I do think this is very important that this becomes part of the normal practice.
I totally agree. That was exactly what I was going to say. Michelle, you and Amber and Julia, your voice is so important for this process, because these are people, the regulators work in a very sterile environment. They don't have that understanding, that perspective, and your ability to translate that information in a way that makes it meaningful is what's driving this. So that's great.
And that's why we're making those videos and, trying to raise awareness with not just regulators, but obviously the general, our communities and society in general about, because like I said, you don't often see these kids, or like Tristan still at 3, it soon won't be, but I can take him out in a stroller and he looks pretty typical. People around us would not necessarily have any clue about what's going on behind closed doors. And I know obviously Julia has done a lot to raise awareness with her blog in deep, in-depth narratives of what goes on with rare disease families. Just coming back briefly to the risk-benefit analysis, we have so many bottlenecks with rare disease. We have funding, we have patient recruitment. One that I'm curious about is physician and clinician willingness to take and Winston, you addressed it a bit in your discussion about engaging institutions, but as you said, most physicians are not necessarily used to this space. And so how can we engage? So a personal anecdote, we try after a year of in vitro studies, consulting everyone and their brother, medical testing baselines, developmental testing baselines, and working with a channelopathy specialist who was had, we had specifically gone as a second opinion to work on these investigational treatments. At the end of the day, after a year, she got cold feet about prescribing off-label a 7-year-old generic drug that is used in children for bedwetting on label. I mean, you can hardly imagine something more benign than that. And so I'm just curious about how can we recruit and provide access, because a lot of us are lucky to live in major cities, but especially for people outside of major cities and even here, and I think connected very well to the community, it was really, really hard to get something as simple as an off-label small molecule prescribed at the end of the day. So how do we help clinicians become comfortable, and how do you guys think about the risk-benefit analysis?
That's a very tough question. My wife is a clinician, and she's, a general practitioner. She sees a lot of patients. It's a lot of sort of workflow. How many, there isn't a lot of individual care. It's kind of well, I mean, she's a good physician, but she's kind of manages a lot of people. So it's one of those things where it's a balancing act for how much you can dedicate as far as time goes.
Yeah.
And it's that yeah. I mean, I'm just picturing the scenario of you approaching her and just like what would actually go through.
Right. They're like, all this risk and extra work. That's an easy no.
Yeah, exactly. It's a challenge. Yeah, you're right. And that's the thing, is how do you mitigate that risk? How do you build a framework that they can operate in, where they can feel comfortable and sort of feel like they're helping? Because she often, obviously, I mean, a lot of physicians are in the business because they want to help and they want to be a part of the solution. But it's a balancing act between feeling like they're going to be a problem and part of the solution. And so how do you put that back into, how do you make that so that they're part of the solution with a little less risk, so that's tough. Yeah, that's really tough. I think,
sorry. Go ahead.
Yeah. Well, I was just going to say, I think that if they're interested in the research like Tim, and you have somebody who's motivated by that aspect of it, that's your best bet. I mean, that's an extraordinary person to find, right? And so we just have to wire them up and make sure they can connect to the world.
I mean, I listened to the FDA Rare Disease Day webinar, at least some of it yesterday. And the afternoon session that I didn't really listen carefully to was about educating medical students about rare diseases. And I think that's maybe an important feature, actually, that, I don't know how good this will be. As I said, I didn't listen to everything that was said there, but I think it's a good initiative to maybe not only teach about like these big diseases that we maybe need to understand as doctors, but also more like generally, what are general features when you deal with a rare disease? Because to some extent is sort of the treatment, but I would also say what we see is this incredibly long diagnostic odyssey. And this is also something that just sort of thinking that if it's not like some of the diseases that I can think of, what do I do? I think in the time of genomic sequencing, bringing down the cost. I mean, I think this is very important for us also as a community to push forward to do this much more quickly. Yeah. And so maybe this will be also part of the education. And then I don't want to sort of ask Winston and Tim and others to sort of do this, but I think, you as experienced clinician with N of ones, teaching your own peers, like, I think taking away some of the fears maybe for them, because literally, I don't think that everyone would be willing to inject an ASO that has never been given to anyone intrathecally. So if we want to roll this out, we need to help these physicians to dare to do it.
So I agree with everything. That there's so many good points that were brought up, and I'm in Jeff's shoes of just like nodding along and agreeing and echoing. So one of the things that I think about is related to both Arena's point. So there's like a long term and there's like the medium term that I think about to add on to this, right? I think in the medium term, we need to not rely on the extraordinary of like finding a person like Tim, and we need to find ways of actually breaking out the different components of Tim's expertise that he brought, and all the other investigators. I would feel so much more confident as a physician. I'm not a clinician, by the way, but I would feel so much more confident, and I could see, my partner, my friends, doing this if said, okay, Jeff at La Jolla Labs with all of his experience helped design this ASO so I know it's safe, right? And that is a compartmentalized thing that Tim had to take on in the beginning, but we should actually parse out. So it's like, the industry knowledge is really benefiting, right? That Nian Wei at IXLs help us screen this in a way that makes sure our target engagement is good. That, we had Charles River and we did all this safety studies. And then I think about, okay, so if we can compartmentalize Tim's knowledge and make it so it's not like finding the extraordinary and making sure that we can, tap into that, that's one part of that medium-term solution. But then there needs to be like a quarterback for that process, right? And this is where we get to the education and the, my personal vision is that there's so many fields in medicine that underwent like a diagnostic or like, observation to intervention transition, right? Like cardiology used to be just not doing too much besides giving some medications, but now they go in and they stent open closed arteries and they deploy new valves and stuff. And genetics is due for that, right? You have everyone staring at, Punnett squares and pedigrees and now like exome sequences. What if they could say, we're going to build a whole branch of, new medical society or a new like fellowship of interventional genetics. And this is where you get to work with the Jeffs, the Arenas, the Nian Weis, and figure out this path so that you can know the pieces that go into ultimately treating that. The key there is that you don't replace your primary care physician, right? Like these are just like often the surgeons who have that unique knowledge. They know what tools to use and they can drop in and they'll follow up. But there's always going to be a primary neurologist or a primary, care doctor who follows that longitudinal care. You have a specialized person who can drop in with that knowledge. So again, I've like thought a lot about the same problems there. And I think there's a big picture way hopefully we can get to.
Yeah, and I mean, I think it will just shift naturally as this becomes more mainstream and more common. I think we're dealing with a similar mindset shift with the patient community for, everyone whose kid is over, say, like 10 or something, who spent most of their kid's life having no clue the basis of their disease. And now suddenly, we're saying, oh, when throughout all of human history, intellectual disability was just, that's completely untreatable, like no questions asked, to like, no, we can actually start to do something about this. Okay, so shifting to kind of some info that would be helpful to us in the rare disease community. Irina, it sounds like you're mostly focused on a particular patient population, but Jeff, say for La Jolla Labs or the work you're doing, how do you decide what disorders to study and treat, and what makes one disease a better candidate than another?
That's a good question. For us, the rate limiting step is actually it is somewhat delivery. So we want to make sure that we can get to the target. But we are primarily a very early discovery company. So we do designs, we do models, and we don't necessarily limit ourselves to anything. We actually have our own N of 1 patients outside of NLORM, but they have to have a physician sponsor and a scientific team to kind of help them understand what's going on. And in some of those cases, we have taken on patients that these are people who basically spill over from NLORM because they were denied from NLORM and ended up in our, coming to us directly. And so we don't. We have some ideas of some things that we want to design that are we have some indications that we want to go after, but those are sort of back burner things that we're kind of looking at. But for the most part, we take on anything that we can get to as far as the ASO is concerned, whether it's a splicing oligo or an SI is another modality that we're using. And then there's all the NLORM. We do all the screens for NLORM. So we have basically 20 patients right now, cell lines in-house.
Okay, very interesting, thank you. And Irina, how about within your specific patient population, same question. How do you decide which disorders?
So, I mean, first of all, we look at the family, the child. We want to initially, because, there is not a process right there that would lead to a rapid approval. So we don't know this yet. So we would like to include initially in the first drug development programs, individuals where we think that we can actually really show a benefit, that we have a high likelihood. I mean, we never know, but that we think so. And also we have to be absolutely as certain as we can be that what we find is the genetic variant is really like the main driver. Like there is not something that modulates this as well. And then of course we want to be sure that we can sort of correct this with an ASO. So while we do consider splice modulation as well as knockdown, like we discussed before, like wild type knockdown. I mean, if there is knockdown, we want to make sure that we think that the level of knockdown we can achieve will actually have a benefit, and that if we cannot deliver a selective ASO, that we still think that whatever we can knock down doesn't pose a risk, but has a benefit. So some of these considerations. And then we also, and we have discussed this with some of the clinicians who are also involved, is like the family, because like, do they understand what it takes, because there is also a burden on the families to come frequently. We want to have certain safety assessments. So just to make sure that as best as we can, that the whole process is agreed upon. And then, I mean, so far, we were approached by individuals, like by physicians. And so far the cases looked like very good, but they have done some prior work on those cases. Once we are more confident that we can deliver this, we can design these ASOs, we have criteria to make sure that these ASOs are whatever, reasonably safe and efficacious. We will broaden these criteria, because then we have actually shown that we can do this. But the initial criteria have to be like that we are really confident, both from the genetic mutation, the delivery as well, and then the stage of the disease and the disease symptoms, that we can show a benefit.
That makes complete sense. Winston, I would love to hear your answer too from your industry side as well. But we're about to have to wrap up. One sentence for me as a takeaway to the patient community. What do you wish for most from patient advocacy groups and the patient community? Winston.
keep on beating the light. Simple as that.
Oh, well, thank you. But concretely, like, what do you need from us?
Oh, I think it's just to me, the data sharing aspect and actually concretely asking for it, right? Saying, you know, may we share this? That would be a huge benefit for I think you don't know how much power you do have in this, and asking for it and then having the community press on this is what the standard is. I think that would be really important.
I loved your point in your talk about not only, you know, obviously publishing is way too slow, but not only like providing info on some platform, but actively seeking out people who might be doing something similar and could benefit from that info. And I think that's something that we can all, you know, work on, because it's easy to get caught up in millions of other things, but taking that extra step to reach out and make those connections could be really huge. Okay. Jeff, in one sentence, and then Irina, one sentence, what do you want from patients?
One sentence. Once again, I follow Winston, exactly what he said. I just very quickly, I want to reiterate that the power of the patient advocacy in saying, this is our data, we want to share it, we want to make it useful. It's there,
Yep.
it's there. You just have to say it.
Okay, good to know. Thank you. And Irina?
And I would just add, continue to lobby, but lobby even more broadly, like the Hill, Congress, like payers. We have to,
Yep.
want that this is not a question anymore. How
Yes.
is this paid for? It is just there. It is a disease that has a treatment. It needs to be reimbursed. I think that's also very important. So just, yeah, I know it's hard, but keep going.
All right. Well, thank you so much. It's really been an honor to get to speak with you all.
Thank you.
Thank you. Thank you.
Thank you.
Bye-bye.
Bye.